Welcome to This Explainer. Look around you. The sheer diversity of life on Earth is literally staggering. But how do scientists actually organize millions of species? Uncover evolution's clever little tricks and trace the very birth of new animals. Well, today we're going to decode the ultimate family tree, the tree of life. Let's get right into it.
Here's our roadmap for today. We'll start by looking at the tree of life and tracing our ancestral roots. Then, we'll see how evolution can totally trick us. Ask what exactly makes a species, and finally, look at how entirely new species form.
All right, Section 1, the tree of life. Let's look at exactly how and why we organize life on Earth.
To make sense of the biological world, scientists use a system called phylogenetics. Basically, it's the naming and placing of organisms into taxonomic groups that reflect their common ancestry. Think of it like the ultimate massive filing system. By sorting through similarities until only a few choices remain, we can find exactly what we're looking for. And this is absolutely crucial because it helps us find shared biological systems across species. It even helps us predict how certain diseases might behave in humans, just based on our shared evolutionary history. How cool is that?
This brilliantly illustrates what we call a nested hierarchy. Notice the evolutionary relationships among vertebrates here. We can trace distinct groups, birds, turtles, crocodilians, lizards, and mammals, all the way back along those branches to shared connection points, or nodes. Those nodes, they represent a common ancestor, showing us exactly how different lineages split off over deep time.
Moving on to Section 2, tracing ancestral roots. Because, well, how do we actually figure out who's related to who on that tree in the first place?
The crucial rule to remember here is pretty simple. The absence of a trait is the ancestral state, and the presence of a new trait is derived. Think of it like a blank canvas versus a painted one. Scientists actively look for what we call synapomorphies. That's just a fancy technical term for shared, newly derived characters. These new traits are literally the structural breadcrumbs we use to figure out which species are closest relatives.
To see this in action, check out the fundamental architecture of these arm bones. On the outside, a human arm, a cat leg, a whale flipper, and a bat wing look completely different, right? I mean, they do very different things: grasping, walking, swimming, and flying. But look closely at the highlighted bone structures. The underlying layout is remarkably similar. These are homologous traits. They're shared because they were retained and modified from one single common ancestor. It really proves that evolution is a tinkerer, just using whatever tools are lying around.
Section 3. When looks are deceiving. Because nature absolutely loves a plot twist.
Let's play a quick game. If you look at a bat, a bird, and an insect, they all have wings, right? But which of their wings evolved from the exact same structural ancestor, making them homologous, and which simply evolved independently to solve the exact same problem of needing to fly?
Well, here's the trick. While both the bat and the owl use these wing structures to fly, their wings are actually homoplastic, meaning they appear similar but absolutely not because of common ancestry. Instead, they happen because of convergent evolution. Both of these animals face the exact same ecological pressure, the need to take to the skies, and they independently develop solutions that solve the exact same problem, even though structurally they are totally different.
And we see this homoplasy play out on a global, almost unbelievable scale. Just compare placental mammals in the Americas with marsupials in Australia. Because they occupied similar ecological niches, on completely opposite sides of the world, totally unrelated animals independently evolved identical traits. I mean, a placental mole and a marsupial mole both became burrowers. The flying squirrel and the flying phalanger both became gliders. It is a stunning example of evolution solving the exact same puzzle twice.
Section 4. What is a species? If looks can be so deceiving, it begs a pretty fundamental question.
How do scientists definitively draw the line between one animal and another? Where does one species end and another begin?
Biologically speaking, it all comes down to reproduction. The standard definition is that a species is a population whose members can interbreed and successfully produce viable offspring. Basically, if they can mix their genetics and create healthy babies that can also go on to reproduce, they cross the finish line. They're considered the same species.
Because producing viable offspring is kind of the whole point of biological continuation. Animals develop strict reproductive isolation to avoid mating with other species. Why? Well, because mating outside your species results in wasted resources, wasted time, and wasted opportunities. To prevent this incredibly costly mistake, species evolve powerful recognition barriers. Think highly specific courtship dances, unique pheromones, or even physical incompatibilities.
Section 5. How new species form. Let's see how this isolation actually creates entirely new species over time.
One major way the tree of life branches is through allopatric speciation. This happens when physical geography separates populations, often through a founder effect, where a small group gets cut off and starts a new lineage. A mind-blowing example is the Drusilla flies in Hawaii. There are nearly 1,000 species of these flies on the islands, and an incredible 98% of them are found on only a single island. Geographically isolated by the ocean, they adapted to specific microenvironments and just exploded into hundreds of completely distinct species.
But sometimes populations split while living right on top of each other, sharing the exact same geography. We call this sympatric speciation. This usually happens because of ecological shifts or changes in timing within that shared environment.
Look at this incredible timeline of the Hawthorne fly. It's a remarkable example of sympatric speciation happening right before our eyes. Originally, these flies only used hawthorn trees to mate and lay eggs. But in the 1860s, a population shifted and invaded apple trees. Then, a century later, in the 1960s, they invaded cherry trees. Because apples, cherries, and hawthorns all produce fruit at slightly different times of the year, the flies literally stopped interacting. Living in the exact same area, their biological clocks shifted to match the fruit, diverging into entirely new species in real time. Unbelievable, right?
It's an incredible thought. As our global ecosystems continue to shift, as seasons change, and as species adapt to entirely new niches, it leaves us wondering, what new, bizarre, and beautiful branches are currently splitting off the tree of life right under our noses? The tree of life isn't static. It's constantly growing, shifting, and tricking us as we try to decode it. Thank you so much for joining me for this explainer, and I hope you leave today looking at the natural world just a little bit differently.